Okay, so next chapter is 14. A lot of the things in this chapter I've already hit on, so should go pretty fast. Here we are talking about something called the central dogma of molecular biology. You have to know the central dogma of molecular biology. You have to. DNA codes for RNA, RNA codes for protein, and sometimes, remember we talked about RNA can circle back and code for DNA using reverse transcriptase. You have to know this. Okay? Now, going from DNA to RNA is called transcription. Going from RNA to protein is translation. You have to know those. Okay. Kind of like this. This is how I think of the central dogma of molecular biology. Okay, so you got a hard drive, right? That hard drive's got lots of files on it. Well, let's just say that hard drive is like your genome, and those files are like genes. Okay? You don't want to take that hard drive out. No. That's your master files, your original copies. You don't want to mess with those. So what you do is what? You make that one gene in your genome, you make it on multiple copies of it on little thumb drives, USB drives. Okay? Well, those little USB drives, if you lose them or whatever, who cares? You can replace them. No big deal. Your original copy, though, is safe on your hard drive. Well, in biology, your original gene is safe in your nucleus, and you've made copies of that gene in the form of RNA, like thumb drives. Okay. Those thumb drives can then go and make protein. That's like the printed document. Now that printed document traced back originates from the document on the hard drive. But the protein was not made directly from that. It was made from a copy. Okay? Central dogma of molecular biology. DNA to RNA, which is the hard drive to the thumb drives, RNA to protein, the thumb drive to the printed copy. Okay. Okay, so DNA holds the data for combining amino acids together. You have to know that. You have to know that. DNA, the sequence of DNA codes for the amino acid sequence. Okay. Now the combination of amino acids makes up the primary structure of the protein. We talked about that a few sections ago. Okay. But here's the important thing in the last point. You really have to know this. And the third point too. The sequence of amino acids determines the shape of a protein. Okay? So the function of a protein ultimately traced all the way back is coded for in the DNA sequence. Okay? Very important point. The function of a protein is coded for in the DNA sequence. How is that? The DNA sequence codes for the amino acid sequence. The amino acid sequence determines the shape of a protein. The shape of a protein determines the function of a protein. You connect the dots, and you can trace that back and say that the DNA sequence codes for the function of a protein. Very important point here. Okay, we talked about this before. We have 4 nucleotides, A, T, G, and C. You have to know that A and T bond link, G and C link. Okay. We also talked about codons. We'll talk about those later on, but those are triplets, 3 bases in a row. We'll talk about those later. Okay. So what's transcription? Transcription is a process of going from DNA to RNA. This is that idea of having a file on your hard drive, and you're making a copy of that file on a thumb drive. Here, that file is DNA on the hard drive, and that RNA copy is that file on the thumb drive. So that's transcription. Couple differences with DNA and RNA. The most important one here is instead of thymine, there's uracil. Okay, so when we have, if you have to find the complement to RNA from DNA, remember that everywhere there's a thymine, put a uracil in the place of that. Okay, we'll talk about, give some examples later on. Okay, and here is the difference with DNA and RNA. RNA is single strand. See that one line on the right? DNA is double strand, 2 lines. Both have adenine, guanine, cytosine, but in DNA, there's thymine, RNA, there's uracil. 3 types of RNA. You should know the 3 types and their functions. Translation. Translation is the process, we'll talk more about this, of going from mRNA into a sequence of amino acids. And that sequence of amino acids forms the protein. And gene expression is the multi-step process of transcription and translation. And we're going to talk about the regulation of this later on. Okay, but what is gene expression? It's that going from DNA to RNA, then RNA to protein. That's gene expression. You can also say that you can think about the central dogma of molecular biology. Same kind of concept. DNA to RNA, RNA to protein. Or I should say, let me be more specific. DNA is the template, and that will make an RNA copy. Okay? That RNA copy carries the information in its sequence to make protein. That is gene expression. Okay. So, more about transcription. Transcription is going from DNA to RNA. Okay, think of one long strand of DNA, right? And we split that strand open, kind of like we did for DNA replication. We split that strand open, and we're going to make a complement. We're going to make a copy of RNA based on one of those strands of DNA. Oh, by the way, you should know what complementary means. I've been saying complementary. That just means they follow the base pairing rules. You should know that. So let's just say that DNA copy. Okay, so we have A and B, right? DNA template, new DNA strand. If we were to separate that DNA template and the new DNA strand and make RNA from the DNA template, look what happens. It's complementary to the DNA. The only difference is uracil and thymine. Okay, where there should be a T, there's a U. Okay, know the difference. If I was to give you a DNA template like this on the test, you should be able to tell me the RNA transcript. Okay? Okay, 2 things here. Transcription, and we talked about this a little bit before. I want to show you this one. Transcription, or the making of RNA, is done by a molecule, a big protein called RNA polymerase. And that's going to do the copying. Okay, that molecule does the copying. Okay. You see this strand? It says RNA transcript. What made that RNA transcript was a big RNA polymerase moving down that DNA template, left to right. And as it moved down, it assembled that RNA transcript underneath. That's what RNA polymerase does. Now, RNA polymerase doesn't really know where to go. Okay? It has to be told. where to go. How it does that is there's a specific region on the DNA we call a promoter, and that comes before the gene. And before that gene, the promoter will recruit, draw in, pull in the RNA polymerase and allow for the production of RNA. Okay, so think of that promoter as like a sign before a gene that says, come over here, RNA polymerase, you need to do your work starting here. That's what a promoter does. Know what a promoter does. A promoter is a region of DNA that will pull in an RNA polymerase and allow it to make RNA past where it grabs it on. Does that make sense? So we just imagine a gene. Okay, we got a gene, a stretch of DNA. Kind of in that picture, see where it says gene region? In that picture below here? And we want to make a copy of that gene region in the form of RNA. RNA polymerase is the molecule we need. But RNA polymerase doesn't know where to go exactly. So there is a small region of DNA before the gene, or in this picture to the left of the gene. And that's going to pull in that RNA polymerase and allow it to physically sit on the DNA molecule. And as it sits there, is going to go from left to right. And that's shown on the right part of that picture at the bottom. And as it goes left to right, it's pulling apart the DNA. And as it pulls it apart, it's going to make RNA, an RNA copy from a strand of that DNA. That's why you see that brown wave type of thing, and then it's associated with that one line of blue. Well, that one line of blue is single-stranded DNA. That brown wave is RNA, and those are complementary to each other. Okay. So just know what a promoter is and what an RNA polymerase does, and know their relationship. Okay, so now we're going to talk about the idea of codons and introns and exons. Okay. So in bacteria, the whole gene is made into RNA. Okay. But in eukaryotic organisms like us, we have the idea of introns and exons. Introns are interruptions, okay? Things that don't code for protein that are in between the exons, and those exons code for protein. Now, what's the point of this? Why do we have our DNA with introns and exons, and we have RNA, and we have to go in and physically pull those introns out of the RNA and connect together the remaining exons to make our mRNA? Why do that? It looks wasteful, doesn't it? Right? It's like you got all this stuff and you got to go through all these extra steps, and it doesn't make sense. Well, it does, though. It really does. We talked about this in class with the amount of genes. We have far fewer genes than we thought we did. Okay. The reason we have fewer genes is because of the idea of alternative splicing, and you need to know this idea of alternative splicing. I'll give several examples in class. Basically what it does is what? I have one gene. That's that light blue, dark blue at the top. I can put together different exons and make different mRNA strands based on selectively pulling out an intron and an exon. So what do I mean by that? Okay, so let's start at the top. We have exon, intron, exon, intron, exon. And it's all under that banner of a gene, right? Okay, let's name that exon 1, exon 2, exon 3. Those two introns there, let's call that intron 1 and intron 2. Okay. And shown in the picture, we pulled out intron 1 and intron 2, but we kept exon 1, exon 2, and exon 3. Okay. What if we got rid of exon 2 and put together exon 1 and exon 3? Can we do that? Sure. That's alternative splicing. We can combine, mix and match those exons. So we can put exon 1 and exon 3 together. Well, could we put exon 1 and exon 2 together? Sure. We'll put exon 1 and exon 2 together and leave out exon 3. Okay. Can we put exon 2 and 3 together? Sure. Not a problem. We'll just take out the exon 1 and that intron 1. We'll take that intron 2 out, but we'll keep that middle exon and that last exon. So what does that do? We're making multiple versions of mRNA based on selectively removing exons and keeping exons. That allows one stretch of DNA to code for many different versions of mRNA. So it's kind of like a compression of information. Okay. After mRNA is made, two things are added. We have a 5' cap and a poly A tail. And this is how it is. It's a bunch of A's at the end. A. Key point here, what is alternative splicing? It's the combining of exons together in different patterns to make different mRNA. That's the first thing you need to know. And then second thing, what is an intron? An intron are interruptions. They are removed. They do not code for protein. What are exons? Those have the capacity to code for protein. Those are retained in the mRNA. Now, not all exons are retained because of alternative splicing. But just know what introns, exons, and alternative splicing are. Now we're going to hit on the idea of the codon. Okay. This is the core principle of translation. Translation is going from mRNA to protein. Okay. A codon is 3 mRNA or 3 nucleotides on an mRNA strand that code for a specific amino acid. Okay. For example, CCU, that codes for proline, an amino acid. GAG, that codes for glutamic acid, an amino acid. Okay. AAG, lysine. What's the take-home point here? Every 3 bases is a triplet, or codon. And one codon codes for 1 amino acid. One codon codes for 1 amino acid. Okay? Very important point here. Why is that important? Well, we only have 4 different types of bases, right? ATGC for DNA, AUGC for RNA. But there's 20 plus amino acids, so how do we go from 4 bases to 20 plus amino acids? Well, if we have 3 bases per amino acid, you do the math, there can be 64 different combinations. And that's the threading of the needle, so to speak. That explains how we go from mRNA to amino acids. That every 3 nucleotides on mRNA, and as a consequence DNA, code for 1 amino acid. So what's the take-home point here? What's a codon? A codon is 3 nucleotides that code for 1 amino acid. That's the take-home point. Okay. Kind of what I said before, there's multiple mRNA codons. Okay. Now some codons, we call them start and stop codons. Start codon is the start of the gene. Usually 99% of the time, it's methionine. Not always, but most of the time. There's also codons that will stop protein synthesis. When you're done with making the protein, you got to stop it somehow. And these stop codons do not code for any protein. They're kind of like dummy molecules. They don't code for any amino acid, so when they are incorporated, it cuts off protein synthesis. We talked about that genetic table. You should have kind of an idea of how to do this. You've got 3 parts: first base, second base, and third base. And it's going to convert a triplet into a particular amino acid. For example, let's say we have, let's go to this one, proline, CCU. Okay? So remember CCU. The first base is C, the second base is C, the third base is U. Okay. So the first base is C. So we go to the left, and it says first base, and the second block is C. Okay? So then we move to the second, and it says the second base, UCAG, second base at the top. And we kind of go on where those two meet. And then we go to the right of that, and it says UAGA, and ours was what? CCU? So we go C, C, U, and see how they all code for what? PRO, proline. Okay. tRNA. Now, how does it? We go to here, right? We have an amino acid in our mRNA. How do we make that connection? Well, how we make that connection is tRNA. tRNA is like a translator, right? It takes one language and converts it to another language. So tRNA is a molecule, and one part of the molecule there will be a specific amino acid. Also on the molecule is what we call an anticodon. Anticodon matches to the codon on the RNA. Okay. One anticodon only links to one amino acid. One anticodon links to one amino acid. For example, ACC will only link to tryptophan on tRNA. AAC will not code for any, or will not have any other amino acid on it. Okay. Ribosomes are the proteins that hold it all together, and these are what is going to help do the assembly. Translation. Translation is going to use what? The tRNA, the ribosomes, and the mRNA, and going to convert from mRNA to protein. So let me just show a picture of this here. Okay. Oh, by the way, translation happens in the cytoplasm and happens in 3 steps: initiation, elongation, and termination. So let's look at the steps. So the first thing is initiation. Initiation is what? The charged tRNA comes in, and that tRNA on one part has an amino acid, and on the other part has an anticodon. Okay. Let's look at, it says initiation, right? And we have an mRNA at the top, and then another mRNA in the middle on this picture. Look at that picture. It says AUG. Okay. That is a codon for methionine. On the tRNA, there's a complement, UAC. Now look at the end of that tRNA. We have a methionine, MET, which is an amino acid. With that anticodon on the tRNA, that thing in the middle, you will not have any other amino acid. It's always going to be methionine. That is the translation. That's the translator. It converts this codon on the mRNA to a specific amino acid on the end of the tRNA. Initiation, that initiation continues. Every time it continues and adds another amino acid, that's elongation. Okay. Continuously adds one amino acid after the next. Adds an amino acid, adds an amino acid, and then adds another amino acid. What's happening here is, look, we're making a strand of amino acids. See that? What? We'll go from here and another slide, and we're adding amino acids in almost like a train or a chain. That strand of amino acids is a protein. Okay. Finally, termination. We hit a stop codon. That long string of amino acids that are connected comes off, and that will be a protein. Okay. So what's the take-home point? Know the 3 steps: initiation, elongation, and then termination. Okay. Then we talked about some mutations, right? Mutations, when you have a mutation in DNA, it carries on to the RNA, and then that can cause a change in the protein. Not always, because a mutation might happen and it might have no effect on the protein. It just depends on the mutation. Okay. We talked about the wobble theory. Okay. So know what a mutation is. A mutation is just a change in the nucleotide sequence. Now that mutation can be a base pair substitution. We'll talk about that in a minute, or a deletion or insertion. So those are examples coming up. But just know what those are. What's a base pair substitution and a deletion or insertion. So a base pair substitution is when another base comes in and takes the place of a base. So for example, let's look at the DNA. Okay, DNA is what? TGA, GGA, CTC, CTC, TTC. Okay. But that T, that middle T, changes from a T to an A. Because that T converted to an A, that is a base pair substitution, what happened? Well, it went from glutamate to valine. See what happened? Because we changed one base in the DNA, the RNA that was made from that DNA also changed a base. Went from A to U. Because it changed the base in that codon, or that triplet, it changed the protein from a glutamate to a valine. That is a base pair substitution. A change in sequence where one base is converted to another base. You can also have an insertion or deletion. Here, one base is removed or one base is added. Major effect here. Everything after an insertion or deletion will be different. Okay, we talked about this. Remember we said the cat and the hat, and if you could only read 3 letters at a time, the cat and the hat, right? But you took out one of those letters. You would read like, if you took out a letter, instead of T-H-E, you would read H-E of the, and then C of cat. So it would be H-E-C. I know I gave that example in class. But what's a deletion or insertion? You are taking out a base, or you are adding a base. And that adding or deleting changes everything past that insertion or deletion, because that the triplet of bases that's encountered past that change is all different. Okay, so just know what a base pair substitution is and a deletion or insertion. Now, the point I'm trying to make here, and you don't need to know all these examples, it's just a point I'm trying to make. You change one base. Okay, that's all you change. One base. Just a conversion or a base pair substitution. One base pair substitution can have, it's like a magic BB in a sense. You change one base, you run the risk, or there's a possibility of changing the entire effect of that protein that DNA codes for. We gave 3 examples, and let me say, there's a magic BB aspect to this. That's what I call it. You could have no effect, but then you could have a major effect. It just depends. It just depends on the situation. Again, it's a case-by-case basis. But we gave 3 examples. Okay. Don't need to know these examples, just the point that major differences can be seen by one amino acid difference. That's all it took. One amino acid difference because of one base difference. That's it. But notice that in these next examples that we changed one base. That one base change changed one amino acid. That one amino acid change changed the whole function of the protein and made a discernible, detectable difference. Okay. Give the one example of people that are of Asian ancestry. Their hair tends to be thicker. The cross-sectional area is about 40% thicker. Why? Well, the current thinking is because it changes an EDAR receptor, and this receptor is important in early embryonic development. Okay. Sickle cell anemia is another example. Again, one base change. Amino acid number 6 is normal, is glutamic acid, and sickle cell is valine. Again, we see a major change. And then cystic fibrosis. Okay, and what causes mutations? 3 things. 1 are transposable elements. We call these jumping genes. There's little segments of the DNA that pull themselves out and go to other places of the DNA. Spontaneous mutations. These are errors that happen in DNA replication. DNA polymerase doesn't work, doesn't get an error out, and it proliferates. Also, harmful environmental agents. UV radiation, cancer, right? If you're going to the sun too much. Chemicals, carcinogens. Okay, environmental agents. It's something called a thymine dimer. 2 thymines that fuse together from too much UV radiation. So we can induce mutations. And again, what are mutations? Small changes in the sequence of DNA. Finally, an important point here is mutations. The only way a mutation can be inherited is if it happens in the germ tissue or your gametes. Okay, you get skin cancer, you don't pass skin cancer on to your children. Now you may pass the tendency for skin cancer on, that's one thing. But if you go out in the sun too much, you don't pass that on. Okay, this change, you have somatic cells and germ cells. Germ cells are egg and sperm. When there's a change in the germ cells, that passes on to the children. That's the only way a mutation passes on. You should know that. Okay, and that wraps up this chapter.